Hello everyone. Welcome to the session of Reproduction in Flowering Plants, Theory Part 4. Disclaimer: the images, text and other material used in our presentations are for educational purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@iitk.ac.in and we shall remove the content immediately. Please note that the part of the content may have been generated using generative AI, which is experimental. So, today's lecture learning objective, what we are going to learn from today's session, to understand the phenomena of pollen-pistil interaction. So, basically, we are going to study about the pollen-pistil interaction. So, in the heading pollination, we will study about the pollen-pistil interaction. And at the end, we will summarize the lecture. So let us understand what is pollen-pistil interaction. Okay. Pollination does not guarantee the transfer of the right type of pollen. What does this mean? Right type of pollen. So the pollen should be compatible pollen. If the pollination has to be successful and later it will germinate and give rise the pollen tube, then it has to be compatible. Otherwise, it will not germinate and the pollen tube will not emerge out. So, for the pollen to be compatible, right, it is very important that they should be compatible. The pollen should be compatible. Then only they will be able to germinate. Otherwise, it will not. Yes? So, what is the meaning of compatible here? First of all, let us understand that. Same species as the stigma. The same species. What does this mean? If the pollination is taking place in the flower between the rows, like one flower is having the anther. Basically, it is a bisexual flower if I am taking the rose example. So, the pollen from one of the flower will be transferred to the stigma of another flower. Right? So, that if it is done in the same species and on the different plant, then the pollen will be compatible. But if it is transferred from the stigma, from the anther of a flower and transferred to the stigma of the same flower, then the gene constitution will be seen. What the gene are having in the case of the pollen grain and what the gene are present in the case of stigma or the pistil part, right? So if they both are having the same gene, then it will be incompatible. It will be incompatible. And pollen will either not germinate, or if it germinate, the pollen tube will not go deep inside the carpel and the successful fertilization will not take place, right? The second thing, if the pollen is transferred from one genus of flower to the another genus of flower, okay, so then also the pollen will be not compatible. So the right type of pollen they are talking about, that the pollen should be compatible, right? So the two things we have discussed here. So often the pollen of the wrong type, either from other species, okay, now here come other species, or from the same plant, and it is self-incompatible. So when it is self-incompatible, so suppose there is a stigma. This is a stigma, and the stigma is having a gene, say S1 type, S1 type, and here the pollen is having, one of the pollen is having S1 type gene and another one is having S2 type of gene, S2 type of gene. So which one will germinate? Which pollen grain will allow to germinate? Actually, the pistil part, the stigma, there is an interaction between the pollen and the pistil, and the pistil allow a particular pollen to germinate on the stigma. So here we see the two types of pollen grain. One is S1 type and another is the S2 type. So which one will germinate here? The S1 will not germinate. It will not allow to germinate here. Only the S2 type will be germinated. Why? The question should be here. Why? Because here the S1 and S1 both are having the same type of genes, right? So it is not incompatible. That is why the term here comes the self-incompatible. Self-incompatible means if the pollen and pistil are having the same alleles of a gene. Actually, this is a gene here which is given as S. Okay, S type of gene, and here you can see S1, S2, S3. Likewise, they could have the number of alleles, right? If they are having S1 and S1 in the case of pollen grain and S1 again in the case of pistil, then they will not allow, the pistil will not allow the pollen grain to germinate, and this comes under the self-incompatible. The self-incompatible means both the alleles of a gene are same and it is not allowing the same type of gene, same type of pollen grain. As they are having the same type of gene, they will not allow to germinate. Whereas here you can see the compatible pollen they are talking about. So S2 is compatible for S1. So this S2 will germinate here. It will germinate and give rise to the pollen tube. Okay. The first thing is here. The first thing is here. And the second thing is that should be of either from the other species or of the same plant. So if they are this one S1 and S1, this pollen grain is from the same plant and S2 is from the another plant. So S2 will germinate, that is compatible. S1 is not germinate, so that is incompatible. And if the pollen grain is coming from another species flower, then also it will be self-incompatible, right? Okay. So the pistil has the ability to recognize the pollen. Very, very important point. The pistil is having the ability to recognize the pollen, whether it is of the right type, means it is compatible, or of the wrong type, that is the incompatible. Now here you can understand what they mean by the right type and a wrong type. Okay, two types of pollen grains are there. One is the right type and another is the wrong type. Now, how they got recognize this pollen? What is the ability of this pistil that it could recognize the right type of pollen and the wrong type of pollen? So, this is very, very important to understand. Recognize the pollen. We will understand. Compatibility protein, yes. There are some chemicals, there are some protein, okay, that are secreted by the pistil, that is the stigma part. Pistil is having a part that is stigma on which the pollen grain comes and rests, right? So this protein and chemicals which are secreted by the stigma is helping to recognize this right type of pollen. Okay, right type of pollen, and the right type of pollen is the compatible one, the one that is having the different allele from the one having in the pistil part, right? Just now I have shown that with the help of a diagram by drawing it, right? Okay. Now, if it is the right type, the pistil accept the pollen and promote post-pollination event that leads to the fertilization. Okay, so if the pollen is of right type, if the pollen is of right type, then pistil accept the pollen. The pistil will allow the pollen to swells up, to germinate, to take out the pollen tube, to penetrate the stigma tissue, to penetrate the style tissue, to penetrate the ovule and lead its way to the embryo sac. It will allow. Otherwise, if it is of wrong type, then pistil will not allow the pollen to germinate. If it get germinated, then also it can be stopped at the stigma level, it can be stopped at the style level, it can be stopped at the ovary level, right? Can pollen and egg of two different species fertilize? No. This was one of the condition. Wrong type. The wrong type is of two things. The first thing, that the pollen is of different species. Second, if they are of the same species, but they should have different alleles, S1 and S2. Suppose the pollen is having the S1 type of gene, then the pistil should be of S2 type if it is right type. But if they are having both the same type of gene, then it will be the wrong type. Okay. So the right type and wrong type understanding is very, very important in the case of pollen pistil interaction. If the pollen is of wrong type, the pistil reject the pollen by preventing the pollen germinate. How it reject? What is the mechanism of rejection? The mechanism of rejection, actually what happened, there are some proteins, there are some signals that are delivered by the pollen grain, and that the pistil also release some chemicals and hormones that judge that protein, what type of protein they are giving out. Okay, the pollen is giving out what type of protein. So according to that interaction, they are able to know whether they are having the same gene or different gene, whether they are coming from the same species or different species, right? So such type of communication or dialogue is there between the pollen grain and the stigma. The dialogue, it means, it doesn't mean that they are talking like we are, that like I am explaining to you and you are listening, you are asking some question in the chat box. It is not the two way like that in the form of dialogue, but yes, there is a dialogue in the form of release of chemicals, release of protein, release of hormones. What is allele? Okay, so we are having the different genes. We are having the different genes in our body. The genes for height, the gene for hair color, the gene for nose, the gene for ear, the gene for blood even, right? And suppose I'm taking an example of blood. So how many blood groups we are having? A blood group, B blood group, AB and O blood group, right? We are having the different form of a gene. The different form of one particular gene is called allele, right? So there is a gene for height, and you know that all the humans are of not the same height. You see, all are of same height? No. Some is having 5 feet, some are 5.5, some are 5.4, some are 6. So gene is one, but the form are different, right? So that is allele. So that is allele. Okay. So here one gene that is for the recognition is there, but it is having the different form S1, S2, S3. So if the the pistil is having S1 type, then it will not accept the S1 type of pollen grain. Understood? Same type of allele will be not accepted. If the pistil is having S1 type and the pollen is having S2 type, S3 type, S4 type, then they will allow to germinate. That is compatible. And if they are having, the pollen is also having the S1 type, then it is incompatible. Right? So that is the right type and wrong type. So in the case of wrong type, we were discussing that there is a dialogue. Dialogue means release of chemicals, and the release of chemicals is actually allowing or not allowing the pollen grain to germinate on the stigma, and further the growth of the pollen tube in the style. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of continuous dialogue between the pollen grain and pistil. See, the term is also written here, that dialogue between the pollen grain and the pistil. What does this mean? Are they talking? No. They are not talking actually, but yes, they are having the communication with each other in the form of release of chemicals, right? So this dialogue is mediated by the chemical component of the pollen interacting with those of the pistil. Okay, so pollen release some chemicals. It is really releasing some chemicals, and the pistil could recognize that the pistil could recognize those chemicals. They can judge whether the pollen is of the same type or of the different type, same species or. So it is a chemical interaction. Yes, it is a chemical interaction. You must have studied about this pollen tube germination, the germination of the pollen grain and coming out of this pollen tube in the class 10th also. In the class 10th, in which topic you have studied? Any idea? Chemotropism. Chemotropism. So there is a release of chemicals, and according to that chemical release, they are having the tropic movement, right? So that comes the chemotropism term there, right? So here again we are talking about the same thing, that the release of chemicals, the dialogue is mediated by the chemical component released by the pollen and also by the pistil. So it is only in the recent years that botanists have been able to identify some of the pollen and pistil components and the interaction leading to the recognition followed by the acceptance or rejection. This is not a very old concept. Recent botanists have came to know that there are release of some chemicals from the pollen as well as the pistil, and with the release of these chemicals, they are able to know each other genes. And on the basis of that, there is an acceptance of the pollen or there is a rejection of the pollen. Right? So this is very important, that there is a chemical integration between the pollen and pistil interaction. So first we will understand what are the different things that happen at in this particular point, pollen pistil interaction. What are the different things that happen? Pollen recognition. First of all, the things that happen is the pollen recognition. The chemical signals between the pollen grain and the stigma initiate recognition. Okay, there is a chemical signal, means there is a release of chemicals. Okay, and pollen grains and the stigma initiate to recognize each other. Second thing, pollen adhesion. The pollen grain adhere to the stigma, facilitated by the proteins and the sugar in the pollen coat and the stigma surface. So there are some protein and sugar that are present at the surface of the pollen grain. That is the pollen coat here. You can see here pollen coat or the pollen surface. And also stigma also release out some sugar material, some sugar substances and protein. Now this protein and this sugar allow the pollen grain to adhere it. Adhere it means get stick to the surface of the stigma. Okay, so it get adhere to the surface of the stigma with the help of the proteins and the sugars released by the pollen coat as well as the stigma surface. And stigma will release this pollen, this proteins and sugar once they recognize the surface, once they recognize the type of pollen tube, whether it is a right type, whether it is a right type or wrong type. Okay, whether it is a right type or wrong type. Pollen tube growth. Upon recognition, pollen grains germinate, forming the pollen tube, and that grow through the style towards the ovary. Okay, once they get recognized, the first thing, once they get recognized, they get adhere to the surface of the stigma. Okay, and after that pollen grains start germinating. They start germinating and they release out the pollen tube from the germ pore. So from where this germ pore, where the germ pores are present? Can anyone tell me where the germ pores are present? On axine. Very good. They are present on the surface of axine. The axine is made up of which component? Next question. The axine is made up of which component? Very good. Sporopollenin. Sporopollenin. The axine is made up of sporopollenin. But at some places, this sporopollenin is very less or absent. Yes? So at the when they are absent at one particular point or 2 or 3 point, so that point is called as germ pore. That point is called as germ pore. And from that point only the germ tube emerge out. The germ tube emerge out. Now, once the germ tube emerge out, it penetrate the stigma. It penetrate the stigma. Right? It penetrate the stigma or style also. It penetrate the style also. Right? And once it reach to the pollen tube, once it reach to the pollen tube, so you asked a question, sir, is intine regular layer? So pollen tube is made up of cellulose, is an extension of the inner wall of the pollen grain, that is the intine. Okay. Yes, it is made up of this intine layer comes out in a form of a tube, and obviously there is a membrane, that is the plasma membrane, just below the intine, right? So this intine layer is involved in the formation of this pollen tube. So pollen tube is made up of cellulose, is an extension of the inner wall of the pollen grain, that is the intine. Okay. This pore is present only on the axine part. This pore is present only on the axine part, not in the intine. The intine comes out. The intine comes out in the form of a tube-like structure. From that pore of the axine only, right? The pores are not present in this intine layer. The intine layer is continuous. It is a continuous layer, right? It is not like the axine that at particular point the sporopollenin is absent. It is not like that. The entire layer is continuous. It is made up of the cellulose and pectin substances. At the point of germ pore, the entire comes out in the form of a tube-like structure. Okay, once it comes out in the form of a tube-like structure, it penetrates the tissue of the stigma, it penetrates the tissue of the style, it penetrates the tissue of the ovary and makes its way toward the embryo sac, the female gametophyte, the embryo sac. Is it clear till here? After that, pollen tube deliver male gamete to the ovule. Pollen tube is having the male gametes in it, and that is delivered inside the ovule, and that is helping in the process of fertilization. Why they are doing so? So that the fertilization could take place, because the male gamete is present in the pollen tube, right? Okay. So as mentioned earlier, the following compatible pollination, the pollen grain germinate on the stigma to produce pollen tube through one of the germ pore. Okay. So here you can see there are so many pollen grains germinating on the stigma. Yesterday, one of the student has asked me that there are so many pollen grains are falls on the stigma and they germinate. But all of it reaches to the embryo sac and do the process of fertilization. No, it is not like that. It is not like that. Though there are many compatible pollen could fall on one stigma. At a time, there can be many compatible pollen grain can fall on the stigma and they all can germinate. They all can germinate. But only one of the pollen tube makes its way earlier and reaches to the embryo sac. Once it reaches to the embryo sac, other pollen tube will cease there. Whether it is at the stigma level, whether it is a style level, whether it is present at the ovary level, they will not grow from there. Okay, once one of the pollen tube reaches earlier to the embryo sac. So here you can see there are so many pollen grains germinating on the stigma, but only one of it will reach to the embryo sac. The content of the pollen grain move into the pollen tube. Okay. What happens? The content which is present in this pollen grain, the content, that is the generative cells. Okay, the generative cells divide, the nucleus of the generative cells divide into the two male nuclei. So the stigma accept only one pollen grain? No, the stigma allow, if suppose 5 pollen grains are there and out of 5, 4 are compatible, so it will allow the 4 pollen grain to germinate. It will allow the 4 because it will release the chemical that is helping for the pollen grain to germinate, right? But only one of the pollen tube will makes its way and leads to early to the embryo sac level. Yes, if it is having the one ovule, if it is having one ovule, then there will be one pollen tube reaches there and it will fertilize one of the ovule, right? But if it is in the case of multiple ovules like papaya, yes, in the papaya you can see the number of seeds are there, right? So multiple ovules. So yes, in that case, multiple pollen tube could reach to the multiple ovules, right? In that case it happened. But for one particular ovule, there will be only one pollen tube that emerges inside the embryo sac. Is it clear? Okay. So the content of the pollen grain move into the pollen tube. So pollen tube, they are having the cytoplasm, they are having the generative nucleus, and this generative nucleus divides into the two nuclei, and that two nuclei are called as male nuclei. That two nuclei are called as male nuclei, right? So the pollen tube grows through the tissue of the stigma and style and reaches the ovary. So if this pollen grain is compatible, then the stigma allow to penetrate, the style allow to penetrate, the ovary allow to penetrate. But if in case the pollen grain is not compatible one and it got germinate somehow it get germinated, then it will be stopped at the stigma level. Suppose it can penetrate the stigma, it will be stopped at the style level. Suppose it penetrate the style level, it is stopped at the ovary level. So there are in different species, there are different mechanism to stop the incompatible pollen grain to leads to the fertilization. There are different mechanism. In some plants, they do not allow the germinate. In somehow they get germinated, then they will be stopped. The pollen tube growth will be stopped at the stigma level or style level or ovary level, right? So there are the different mechanism to stop incompatible pollen grain. But if it is compatible pollen grain and the pollen tube emerges out, so it will easily allow to penetrate the stigma, penetrate the tissue of a stigma, penetrate the tissue of a style, and penetrate the tissue of ovary and reaches to the ovule part, embryo sac. So now you can understand why this compatibility is so very important. To understand this particular term, compatible and incompatible. Okay. So here you can see this diagram is very, very important to understand. Here you can see there is just one ovule is there. Okay. One ovule is there. And here you can see the number of pollen grain. This pollen grain is not germinating. This pollen grain is not germinating. So it may be incompatible. So this pollen grain, the three are germinating here and the two are not germinating. So all these three are not three, actually the four. The four are there and the four are germinating. It may be the compatible one. Pollen tube grow in case of self incompatible, in case of outbreeding device. So if it is self incompatible, if pollen is incompatible and it somehow germinate, then fruit will be healthy to eat. See, one thing, just clear it. The fruit formation will take place only when there is a fertilization. The ovary changes to fruit only after fertilization. The pollen tube germinate. The pollen germinate and give rise to the pollen tube. But if somehow the fertilization doesn't take place, so the fruit will not form. Okay? The fruit will not form. After fertilization only, the ovary changes to fruit, the ovule changes to seed. So you are saying that if pollen is incompatible and it somehow germinate, then the fruit will be healthy to eat. If it is incompatible, then at some particular mechanism, it will not allow the fertilization to take place. So there will be no fruit formation. Right? Okay. So here you can see only one of the pollen tube is reaching to the ovule part. Only one of the pollen tube is reaching to the ovule part. And you can see this particular diagram, this hair-like structure, these are what? pollen tube growing through the style. There are so many pollen tube. There are so many pollen tube. So you can think about the number of the pollen grains that are present at the stigma. Right? So you would recall that in some plant, the pollen grains are shed at the two-celled condition, a vegetative cell and a generative cell. This we have already studied in the male gametophyte when we are studying about the first lecture, that we discussed about that the pollen grain is of two-celled stage. In the 60% of the angiospermic plant, the pollen grains are shed off. Shed off means they are released from the anther at two-celled condition. One cell is vegetative cell and one is the generative cell. Clear? Is it clear? Okay. Now, the second condition, the rest of the plant. What is the rest of the plant? 40% of the angiospermic plant shed off their pollen grain at three-celled stage. How this three-celled stage is coming? There is one generative and one vegetative cell. Now this generative cells divide itself in the two cells. And when they are present in the anther only, and then they are released out. So in the 40% of the one generative cell divide, yes, there is one generative cell and one vegetative cell, but this generative cell divides into the two cells. That is the male nuclei. They are having the nucleus more and very less amount of the cytoplasm, because you all know that the generative cell is having a very small spindle shaped and it is having very less cytoplasm, and when it divide, the cell divide, it will have again a very less amount of the cytoplasm. Sorry. Now here, the two-celled condition, a vegetative cell and the generative cell, majority of the flowering plant is shed off at the two-celled condition. Now in such plant, the generative cell divide and form the two male gamete during the pollen tube. When there is a growth in the pollen tube at that time, they are talking about the two-celled condition. The pollen grain that is shed off at the two-celled condition. Now, in a plant that is shed the pollen in the three-celled condition, the pollen tube carry the two male gamete from the beginning only. Because they are already having the two male gametes. But the one that is shed off at the two-celled stage, the pollen grain that is shed off at the two-celled stage, the two male nuclei is formed when there is a growth of pollen tube in the stigma. At that time, the generative cell divide into the two male nuclei. So this is the difference. The pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enter one of the synergid through the filiform apparatus. Right? Pollen tube, after reaching the ovary, when they are reached to the ovary, they enter the ovule through the micropyle. There is a micropylar pore. Pore means a hole-like structure. Sir, what is the ploidy level of the two-celled and the three-celled stage? Okay, you are talking about the ploidy level. What is the ploidy of the microspore or the pollen grain? Can you tell me? Pollen grain. Recall how the pollen grain has been formed. It is N. N means haploid. So the haploid cell when divide, it will again form haploid. See, haploid cell is dividing in two cells. So it is the mitotic division. It is the simple mitotic division. Right? The meiotic division or the reduction division has already been taken place in the formation of the microspores. The microspore tetrad we have talked about. The four pollen grains are intact together. Their walls are sticked together and they are in the form of tetrad. But actually there are the four cells, the four pollen grains. When they get mature, they are all dehydrated, they all are released into the 4 pollen grain. And that pollen grain we are talking about. So pollen grain is itself a haploid nucleus. And when its nucleus divide mitotically, then it will be 2 haploid nucleus, right? Okay. So the ploidy level of the 2-celled in the, that is the generative cell and vegetative cell, both are haploid. The 2 male nuclei which is formed here by the division of the generative cell is again haploid, right? Okay. See, this particular line is very important. Pollen tube after reaching the ovary enters the ovule through the micropyle and then enters one of the synergid through the filiform apparatus. So two things you must remember here, the micropyle. Why this micropyle is there? Micropylar pore is there so that the pollen tube could enter through this particular point and enter one of the synergid. When it reaches to the embryo sac level, so it get enter inside one of the synergid. Actually there are the 2 synergid, so it can enter any of the one of the synergid. And in the synergid there are one structure that is called as filiform apparatus. So this filiform apparatus play a role in guiding the pollen tube towards the embryo sac during the fertilization. It is allowing the pollen tube to get enter through this filiform apparatus. It is actually guiding this pollen tube to come and enter through my cell. I'm allowing you to come enter the synergid cell through these filiform apparatus. And once they get enter inside this pollen tube, get enter, penetrate the cell and get enter inside the synergid cell, it releases all its content there in the synergid cell. What is the content of the pollen tube? Some sort of cytoplasm and the 2 male nuclei or the 2 male gamete. That is the content. It is released in the synergid cell. It is released in the synergid cell. And this is the function of the synergid cell, to receive the pollen tube, to receive the content of the pollen tube. What is the function of the filiform apparatus? It helps to guide the pollen tube and makes its way toward the synergid cell, right? So it helps to ensure the successful fertilization by directing the growth of the pollen tube towards the female gamete's egg for the fertilization to occur. Are 2 male nuclei and gamete are the same thing? Yes, they are same thing. They are the same thing. Okay. The 2 male nuclei, or you can say the male gamete, you are saying the same thing. There's no difference. 2 male gamete or 2 male nuclei. Same thing. Okay. So don't get confused with these two terms. They are having the same meaning. So it helps. Which is helping here? The synergid cell. It helps to ensure the successful fertilization by directing the growth of the pollen tube towards the female gamete's egg for the fertilization to occur. Sir, N in ploidy is no of number of chromosomes or amount of DNA. We are talking about the number of chromosomes. We are not talking about the amount of DNA. When you are talking about the ploidy, then you are considering the number of chromosomes. Ploidy means the number of chromosomes. So have you thought about, sometimes we talk about the chromatin and sometimes we talk about the chromosome? See, at the time of division, generally the cell is having the chromatin material, one intact thread-like structure. But at the time of cell division, the cells are broken into small fragments. And those fragments are called what? Chromosomes, right? Because it is very difficult to divide this long stretch of DNA in the form of chromatin fiber. So that fibers are broken in the form of small fragments that are called as chromosomes. And the ploidy number, the ploidy means the number, the repetition of the number, that is that is lead to the this number of chromosome only. So if you are saying the diploid, triploid, haploid, then you are talking about the ploidy of that chromosome. So the N is the basic, that is the half of the diploid, basic chromosome number. So what is the basic chromosome number of human being? Can anyone tell me? Basic chromosome number of human being. Anyone? 2346, how it could be? It could be either 23 or 46. No, 23 pair is again incorrect. It is 23. Half of the diploid. Half of the diploid. That is N. That is N. So, either you are saying 23 pair, then again it means 46. And if you are saying 46, then it is 46 chromosome. Because every chromosome is in the pair formed, right? So 23 pair means, and 23 pair you can say only in the case of female one, not in the case of male, because the this last chromosome, 23rd one, it is having X and Y. They are different types. Okay? Okay. So just think about ploidy we are talking about. Then ploidy means the repetition of the basic chromosome number. N is the basic, that is the haploid. Then 2N means double of it, triple of it. Okay? Like this way. Right? Coming back. It helps to ensure the successful fertilization by directing the growth of the pollen tube towards the female gamete. Okay, so without this directing of the synergid cells, do you think the fertilization could be possible? I don't think so. Because this is a very, very important significance of the synergid cell. And sometime it could be asked, what is the importance of synergid cell in your examination? Filiform apparatus guiding the pollen tube to get enter inside the synergid cells, right? So this is very important. Don't forget this. So here you can see this filiform apparatus allowing this germ, this pollen tube to get enter inside this. It is guiding it. And here the pollen tube releases all its chemical. You can see the male gamete or male nuclei. The same thing. Okay? The male gamete or the male nuclei is the same thing. And here you can see one male nuclei is here and one male nuclei is here, right? So all these events from the pollen deposition on the stigma until pollen tubes enter the ovule are together referred to as pollen-pistil interaction. So from where to where? Why we have discussed so much about the pollen tube germination, pollen tube emergence in the stigma, in the style, in the ovary, and then getting enter inside the embryo sac? Because till here it is the part of the pollen-pistil interaction, right? Pollen deposition. The first thing that happen in this pollen-pistil interaction is the pollen deposition. Pollen grains are deposited on the stigma of the pistil. One is going to the polar nuclei and one to the egg. Very good. Very clear. But this double fertilization will study tomorrow in the next session. Okay. See here. The first thing here happened that in the pollen-pistil interaction is the pollen deposition. The pollen grain are deposited on the stigma of the pistil. Second, pollen recognition. The chemical signals between the pollen grain and the pistil initiate the recognition. Okay. The third thing, pollen adhesion. Adhesion means the pollen grain get stick to the surface of the stigma because of the release of some sugar and the protein material, right? And pollen germination. Germination of the pollen grain occur forming the pollen tube. As pointed out earlier, the pollen-pistil interaction pollen-pistil interaction is a dynamic process involving the pollen recognition followed by the promotion or the inhibition of the pollen. Okay. So we have talked about this in the pollen-pistil interaction, that this is a dynamic process. Dynamic process means it is a continuous process. It is not static. It is moving always. Okay. Dynamic process involving the pollen recognition followed by the promotion or inhibition of the pollen tube. The knowledge gained in this area would help the plant breeder in manipulating the pollen-pistil interaction even in the incompatible pollination to get the desired hybrids. So, this understanding is very, very important for the plant breeder as well, because when there is a rejection at the level of pollen-pistil interaction, but they want somehow that there should be the fertilization process to happen. So they could overcome this incompatible pollination by use of some chemicals, some hormones, and they could get the desired hybrids with that. They could get the desired hybrids. So you can easily study the pollen germination by dusting some of the pollen from the flowers such as pea, chickpea, Crotalaria gentiana, balsam and vinca on a glass slides containing a drop of sugar solution. You can make a 10% sugar solution and you can take that drop of sugar solution on the glass slide and put some of the pollen grain like from the pea plant, chickpea plant or Crotalaria plant. Okay. And you can see what happens under the microscope. So after about 15 to 30 minutes, you will observe that the slide under the low power lens of the microscope, you can see that after 15 to 30 minutes, the pollen tube is emerging out. What is the function of sugar solution? Actually, this sugar solution allow, because this sugar and protein are given by the stigma also. But here you are allowing this pollen tube, pollen grain to germinate artificially. So you should give some sugar so that this pollen tube could germinate. Pollen could germinate and give rise this pollen tube. So that's why you are using this sugar solution. In natural way, this is provided by the stigma, but here there is no stigma because you are doing this on the experimental basis. You want to see how the pollen tube looks like, right? So you're likely to see the pollen tube coming out of the pollen grains. A breeder is interested in crossing different species and often genera to combine desirable characters to produce commercially superior varieties, right? So the breeders are interested to crossing the different species of often the genera, different genera also, like, yes. So they want to do this and they could do with the use of some chemicals, some hormones. Genera, like we are having the two genera, Panthera tigris, Panthera leo. So here this is the scientific name of tiger and lion. So Panthera is genus and Leo is the specific epithet, species name, right? So Panthera here, both Panthera tigris and Panthera leo. The term Panthera is the genus. So artificial hybridization is one of the major approaches for the crop improvement program. So in such crossing experiment, it is important to make sure that only the desired pollen grains are used for the pollination and the stigma is protected for the contamination. So here what we can do the process of emasculation. We can take a flower and we can take out all the anthers from it and make it a female flower. All the anthers are removed out, then it will be the female flower and you can bag it. You can bag it so that another pollen grain could not come here and germinate on the stigma. So that is the bagging process. So if the flower parent bears bisexual flowers and remove the anthers, you remove the anther with the help of the forceps. Okay. So you are making that flower as a female flower. So removing of this anther is called as emasculation. Yes, the bagging means covering stigma so that you could able to put some desired pollen on the stigma, that stigma. Otherwise, stigma, the pollen grain from outside can come on the stigma, and this whole experiment will of no use. So this step is referred to as emasculation. Emasculation. Emasculated flower have to be covered with the bag of the suitable size, generally made up of butter paper. So it is made up of butter paper, and that prevent the contamination of the stigma with unwanted pollen, so that unwanted pollen could not come there on the stigma and do the process of pollination. So this process is called bagging. Immediately after the emasculation, you put this bag over it. When the stigma of the bagged flower attain the receptivity, mature pollen grain collected from the anther of the male parent and are dusted on the stigma, and the flowers are rebagged again and the fruits allowed to develop. Can we cover it with the plastic bag? Yes, some plastic bags are also used. It can be used. Okay. So later on you can take the desired pollen grain, and when the stigma is mature, become receptive, then that pollen grain could be dusted on it and it is again covered, right? So if the female parent produces unisexual flower, there is no need for emasculation. If there is a unisexual flower, that is male flower is different, female is different, then there is no need for the emasculation process. The female flower bud are bagged before the flowers open. And when the stigma become receptive, pollination is carried out using the desired pollen grains after flower rebagged. So now we will summarize it, the today's session. Pollen-pistil interaction is the crucial process in the plant reproduction, and the pollen grains land on the stigma, germinate there and grow through the pistil's tissue and reach to the ovule for the process of fertilization. This interaction involves the complex molecular recognition system, including the cell surface protein and the signaling molecules that ensures the compatibility between the pollen and pistil. This we have studied in detail today. The process includes multiple steps: pollen adhesion, hydration, germination, and pollen tube growth through the stigma and style. Can we use bagging technique for the anther also? There is no need for doing for the anther because stigma is never moved. Okay, stigma is never moved. The pollen grain move. Okay, so it is generally done for the female flower. So during this journey, the pistil provide necessary nutrients and the guidance cues to the growing pollen tube, while also maintaining the barriers against the incompatible pollen to prevent the self-fertilization in many species. But yes, sometime I think that the bagging could be done for the anther also, because some other pollen could come and fall on the anther. And when you collect the anther from that flower, you can collect some other pollen that has been fallen on that particular anther. Yes, this could happen. The chances are very less, but this could happen. Okay, so we have completed today's session. Okay, we will have this DPP now. So here comes the first question. In which of the following condition autogamy is prevented but not the gatineogamy? The autogamy is prevented but not the gatineogamy. So the options are as follows. A. Self-incompatibility of a pollen and stigma. B. Non-synchronization of the pollen and the anther release. C. Monoecious condition. And D. Dioecious condition. Yes. So, monoecious condition means both male and female reproduce and give organ as same plant. Okay. So, the correct answer is monoecious condition. Okay. Monoecious condition. Not the dioecious. Okay. So, we will save it and move to the next question. Dioecious means the male and female reproductive organ on the separate individual. Okay, that is not the condition. Okay, the second question is the presence of the physical barrier between the stigma and stamen prevent autogamy. This phenomena is called as the presence of physical barrier between the stigma and stamen. So this phenomena is called as A. Hercogamy, B. Heterostyly, C. Dichogamy, and D. Cleistogamy. So, the option number A, hercogamy, is the correct because in the hermaphrodite flower, the flower which is having both male and female reproductive organ, they are separated within the same flower. They are separated within the same flower. So, hercogamy is the correct answer. We'll save and move to the next question. Third question, flowering plant have developed many devices to discourage the self-pollination and to encourage cross-pollination except they are saying. So options are A. Declining, B. Dichogamy, C. Homogamy, and D. Self-incompatibility. So all the phenomena are here discouraging the self-pollination and encouraging cross-pollination except one. Except one. That is the homogamy. Yes, very important. Good. Homogamy. Homogamy means the anther and the stigma mature at the same time. So when they mature at the same time on the same flower, then there will be the self-pollination, right? So this is the except this homogamy, all are discouraging the self-pollination and encouraging cross-pollination. So we will opt option C, save it and move to the next question. So this is the statement based question. So let us read out the statement A to E. A. Pollination by water is more common among the abiotic pollinating agent. B. Pollination by water is quite rare in the flowering plants. C. Water hyacinth and the water lily, the flowers emerge above the level of water and are pollinated by waters. D. Wind pollinated plants numerous flower packed into an inflorescence, and self-incompatibility is an outbreeding device which prevent inbreeding. So the first option, first statement, pollination by water is more common among the abiotic. No. It is among the abiotic, wind pollination is common. So A statement is incorrect. Pollination by water is quite rare in flowering plant. Yes, it is the rare one. So B statement is correct. C. Water, hyacinth and water lily, the flower emerge above the level of water and are pollinated by water. No, they are pollinated by insects. So C option is incorrect. And wind pollinated plant, numerous flower packed into an inflorescence. Yes, this is the correct one. They are grouped in an inflorescence. And self-incompatibility is an outbreeding device which prevents inbreeding. So, three statement B, D and E are correct. So, we will opt option D, save it and move to the Next, last question. Outbreeding device which prevent both autogamy and geitonogamy is. One of the outbreeding device that prevent both autogamy and geitonogamy. Being monoecious as in date palm. B. Self-incompatibility as in tobacco. C. Being dioecious as in papaya. And D. Being monocliny as in cucumber. So which one is correct? You can tell me in the chat box. Preventing both autogamy and geitonogamy. No. Being dioecious as in papaya. Papaya plant, they are having separate plant, male plant and female plant. On one of the plant, there will be only male flower, and one of the plant, there will be only female flower. So being dioecious as in papaya. So we'll save and submit and see the result. So yes, our all the statement are correct. So this is all about today's session. Thank you very much.